Okay, so welcome to this next video in which we are discussing the opioid receptors. Okay, so what we've seen so far is that there are four different forms of opioid receptors. There is the mu opioid receptor, there is the kappa opioid receptor, there is the delta opioid receptor, and then finally there is the opioid receptor like 1 receptor, the ORL1 receptor. Okay, now all of these are basically coupled to the GI heterotrimeric G protein, which basically means a heterotrimeric G protein with an alpha subunit that is either G alpha I1, G alpha I2, or G alpha I3, but it doesn't give you any information about what the beta and what the gamma subunit are. In addition, there is a little bit of debate as to whether these opioid receptors are also capable of activating heterotrimeric G proteins, which have alpha subunits that are other alpha subunits that are members of the G alpha I/0 family. Okay, so there is some debate over whether some of these other members of the G alpha I/0 family of alpha subunits, if the heterotrimeric G proteins containing them can also be activated by the opioid receptor. But what we do know is that the heterotrimeric G proteins which do have G alpha I1, G alpha I2, or G alpha I3 alpha subunits are most definitely activated by these four forms of opioid receptors. So let's now have a look at this pathway. Okay, so this is the plasma membrane here, and we now have an opioid receptor, and we can draw them all exactly the same way because we know that they are all these 7 transmembrane proteins, and they are all rhodopsin family G protein coupled receptors. So they all bind their ligand in the transmembrane domain region here. Okay? And now what happens after this? And we'll come back to what the endogenous ligand for the opioid receptors is later. Okay, so at the moment you might be wondering, well, why on earth do you have receptors for morphine in the body? The reality is there are endogenous ligands. Okay, you don't just have receptors for morphine. Morphine just happens to be a ligand for these receptors. Okay, they do have endogenous ligands. There are molecules, proteins, which do activate these opioid receptors. Okay, right, so the ligand binds to residues within the 7 membrane-spanning alpha helices, and what that will trigger is a conformational change in the G protein coupled receptor that will be transferred down to these 3 intracellular loops. And what will happen is these intracellular loops will change conformation to produce a binding site for a GI heterotrimeric G protein. So let's put this here. So let's start with our heterotrimeric GI G protein in the off state, which means that this alpha, and I'll call it just alpha I, but what of course I mean is alpha I1 or alpha I2 or alpha I3. Okay, it means that this alpha I subunit has guanosine diphosphate linked to it rather than guanosine triphosphate linked to it. Okay, so it has GDP currently linked there. Okay, and that means that it's currently associated with the beta-gamma complex here. And remember, the gamma subunit of the beta-gamma complex has a prenyl group added onto it. So here is the beta-gamma complex. Okay, so let's colour them in different colours. Okay, so we'll have the alpha I subunit in red here. Okay, and then we'll have the beta-gamma complex. We'll colour in the beta and the gamma subunits in separate colours. So we'll colour in beta in blue, and then gamma in green. Okay, right. Now, basically, what happens is once the ligand has bound to the opioid receptor, whether it's a mu opioid receptor, a kappa opioid receptor, a delta opioid receptor, or an opioid receptor like 1, basically, what will now happen is the intracellular loops will change conformation, and the heterotrimeric G protein will now be able to bind to the intracellular loops. Okay, and specifically the portion which binds is just the alpha portion, so the whole thing doesn't bind, it's just the alpha portion which is going to bind there, the alpha subunit. However, the alpha subunit needs to have the beta-gamma complex associated with it in order for it to be able to bind to the intracellular aspect of this activated G-protein coupled receptor. Okay, so it can't bind unless it's got the beta-gamma complex. And to have the beta-gamma complex present, you need to have guanosine diphosphate bound to the alpha subunit. Okay, so this amounts to only inactive heterotrimeric G proteins being able to bind to the intracellular aspect of the G protein coupled receptor. So, the alpha subunit will go and bind to the intracellular aspect of the of the G protein coupled receptor, and what that will then trigger is it will trigger the alpha subunit to drop its guanosine diphosphate and instead take a molecule of GTP from the cytoplasm. So let's try our best to draw this. Okay, so basically we've got our heterotrimeric we've got our G protein coupled receptor here with its 7 membrane spanning alpha helices. Okay, and I'll try and squash the carboxylic acid tail there and the amino terminus here. And basically now what's happened is this alpha subunit has bound to the G protein coupled receptor here. Okay, and at the present, let's say it's still in the inactive state, and therefore it still has the beta-gamma complex associated with it. Now, the beta-gamma complex, as I say, does not bind to the G protein coupled receptor. However, the beta-gamma complex being bound to the alpha subunit is essential for the alpha subunit to be able to bind to the intracellular aspect of the G protein coupled receptor. Okay, so let's colour this all in then now. So we have the beta subunit in blue here, the gamma subunit in green, and the alpha subunit here in red. Okay, and I'll also colour in to highlight the lipid moieties as well. Okay, so we have our palmitoyl or myristoyl group of the alpha subunit, and we have our prenyl group, which I'll colour in orange, coming off the gamma subunit. Okay, and now what's going to happen is once the alpha subunit is bound to the intracellular aspect of the heterotrimeric G protein, and this binding site is specifically made up of these 3 intracellular loops, what will happen is the alpha subunit will give up its guanosine diphosphate and instead is going to bind a molecule of guanosine triphosphate that it gets from the cytoplasm. Now, the instant this binds guanosine triphosphate, what's going to happen is that the beta-gamma subunit will cleave away from the alpha subunit. Okay, so the instant the alpha subunit binds guanosine diphosphate, and remember this alpha subunit is specifically an alpha I subunit, the instant the alpha I subunit binds the GTP rather than the GDP, then the beta-gamma complex will then break away basically, and now the alpha I subunit cannot bind to the G protein coupled receptor. And I'm sorry, I keep wanting to call the G protein coupled receptor the heterotrimeric G protein. Okay, the instant the beta-gamma complex breaks away, now the alpha I subunit can't bind to the intracellular aspect of the G protein coupled receptor, and it therefore breaks away as well. So what you end up with then is these 2 separate subunits, okay, floating around on the inner leaflet of the phospholipid bilayer on their own. Okay, so here we have alpha I, which is now in the on state with GTP bound to it, and we also have the beta-gamma complex here, which is this assembly of a beta subunit with a gamma subunit. Okay, and I'll put this here. So we have alpha I in red here with GTP bound to it. Okay, and it also still has that palmitoyl group, which is latching it into the inner leaflet of the phospholipid bilayer. And then we have the beta-gamma complex here, beta in blue, gamma in green, okay, and the prenyl group in orange here. Okay, right. And both of these are then going to go on to have signaling effects, basically. So we will begin with the alpha IGTP subunit, but we will go on very quickly to the beta-gamma complex, because actually in the case of opioid receptors, and specifically in the case of analgesia produced by opioid receptors, the beta-gamma complex actually has the more interesting role. Okay, but just for convention's sake, we will start with the alpha IGTP subunit. Okay, so it's quite famous, the alpha IGTP subunit. Most medical students know what this does. It inhibits adenylyl cyclase enzymes, is the answer most people will give you. However, it is slightly more complicated than that, because there are actually a great number of adenylyl cyclase enzymes. So let's introduce the next star then of the show, which is the adenylyl cyclase enzyme. Okay, now adenylyl cyclase enzymes are integral membrane proteins, so they are bound to the cell membrane, basically. Okay, and they have this rather amazing structure. Okay, so here is the amino terminus of the adenylyl cyclase enzyme, which is on the cytoplasmic side of the plasma membrane. So in all my drawings, unless I state otherwise, the top half will be the extracellular fluid and the bottom half will be the cytoplasm. Okay, so you have your first membrane spanning alpha helix here. It then turns around into the second membrane spanning alpha helix, the third, the fourth, the fifth, and then the sixth. Okay, and this cluster of 6 membrane spanning alpha helices is known as the transmembrane domain 1, TMD1. So the same sort of notation as we had for G protein coupled receptors, except that this time it's 6 membrane spanning alpha helices all together that are the transmembrane domain 1. Okay, then you have a loop between transmembrane domain 1 and transmembrane domain 2, which then has 6 membrane spanning alpha helices as well. So transmembrane domain 2 is then a cluster of 6 membrane spanning alpha helices again. And then finally you end up with a little tail called the C-terminal tail again, and then you have the carboxylic acid group of the polypeptide of the adenylyl cyclase enzyme on the cytoplasmic side. Okay, so this is an adenylyl cyclase enzyme. Okay, right. So, basically there are 2 very important domains of this adenylyl cyclase enzyme. You may think that it's the transmembrane domain 1 and the transmembrane domain 2 which are the most important, because they look the most impressive on this picture that I've shown you here. But actually, it's this loop here and this C-terminal tail that are the most important bits. Okay? These bits just latch it onto the membrane. This loop between transmembrane domain 1 and transmembrane domain 2 is called the C1 loop. Okay? This C-terminal tail is called the C2 domain. So we have the C1 domain and the C2 domain. Now they can be divided up further. So C1 can be divided into 2 portions and C2 can be divided into 2 portions. So the first portion of C1, this portion here, is called C1a, and the second portion of C1 is called C1b. Okay? Then C2 has 2 portions as well. The first portion of C2 is called C2a, and the second portion of C2 is called C2b here. Okay, now, which portion of this enzyme is the actual active enzyme? Okay, so what do adenylyl cyclases catalyze? Well, they catalyze a reaction which we'll go through in a moment, where ATP is turned into cyclic AMP and pyrophosphate. So for now, if you don't know what that means, just know that they catalyze some reaction. Now, where is the actual active site? Well, basically, in this picture, it isn't built yet. This is an inactive enzyme, okay? To become active, you need the C1a domain here and the C2a domain to dimerize, basically. You need them to join together, to assemble together, to make an active enzyme which can then catalyze the cyclase reaction. Okay, so to activate the adenylyl cyclase enzyme, what has to happen is C1a has to dimerize with C2a. So let me show this. Here's the plasma membrane, then we have the amino terminal here, Okay, then 6 membrane-spanning alpha helices together as transmembrane domain 1, then this C1 loop here, then these 6 membrane-spanning alpha helices which make transmembrane domain 2, and then the C-terminal tail here. Okay, and I have stretched out the C-terminal tail somewhat so that I can show C1a dimerized with C2a here. So this is C2a, this is C1a. Okay, so to show that I've had to stretch C2a out a little bit. So here's C1a, here's C2a, and they're dimerized together now, and this is now the active enzyme. Okay, so this is what an adenylyl cyclase looks like when it's inactive. This is what they look like when they're active. Now, what do they catalyze when they're active? Well, basically, they catalyze the conversion of ATP, which is adenosine triphosphate. And I will draw you a little cartoon of what adenosine triphosphate looks like. Okay, so firstly, in order to understand the structure of adenosine triphosphate, you need to know what adenosine is. Now, adenosine is the name for adenine bound to a ribose sugar. Okay, so ribose is a pentameric sugar. It has this 5-membered ring with the 5th carbon coming off up here. Okay, so this top member here isn't actually a carbon, it's an oxygen. So you have an oxygen there, then you have 4 carbons within this pentameric ring, and then the 5th one comes off separately up here. Okay, now basically you can bind ribose to organic bases, and if you bind it to the organic base adenine, that then turns, well, the entire molecule now is called adenosine. So adenosine means an adenine organic base bound to a ribose sugar. Okay, so let's colour in the different portions. So I'll colour in the adenine organic base, which I have just shown as a rectangle because we don't need to know what its actual structure is for what we're doing. Okay, and it's quite, well, it's more complicated than a pentagon to draw. You'd have to draw 2 rings next to each other. Okay, so I've just demoted it to being represented as a rectangle for simplicity. Okay, then this is adenosine, adenine plus ribose. We want adenosine triphosphate, so we want to stick 3 phosphate groups off this. Now, I would usually draw them going vertically upwards, but obviously I haven't got enough space, so I'll have to draw them going sideways like this, which now just looks odd, but that's life, okay? So we have these 3 phosphate groups coming off the adenosine molecule now, and that is what is known as adenosine triphosphate. Now, these phosphates are so important, they're all given their own names. So the first phosphate is called the alpha phosphate, the second phosphate is called the beta phosphate, and the third phosphate is called the gamma phosphate, so just the first 3 letters of the Greek alphabet. Okay, right. Now, this is the substrate for these adenylyl cyclase enzymes. So this is what we're going to take in. What are we going to then produce? We're going to produce cyclic AMP and pyrophosphate. Now, basically, pyrophosphate is just 2 phosphate groups linked together. So it's 2 free phosphate groups which have been linked together. Okay, so this is a pyrophosphate molecule here. And I'll colour both of them in purple. Okay, so basically, where are we going to get this from? Well, we're going to cut off the beta and the gamma phosphate here, like so, and that then is called pyrophosphate. The molecule that you then end up with, which is adenine plus ribose, adenosine, with a single phosphate group on, that's adenosine monophosphate, AMP. Now, we want cyclic adenosine monophosphate. So what we're actually going to do is bind this alpha phosphate group to the alcohol group that comes off the 3rd carbon down here. Okay, so let me show you that. So here is our ribose sugar. Here is the adenine organic base off the side. Here's the 5th carbon. And then here's the phosphate group which binds to the 3rd carbon down there. Okay, so let's colour this all in. Here's the phosphate group coming off the 5th carbon. Here is the ribose sugar in blue here. And then finally we've got the adenine organic base, which is in turquoise here. Okay, and this molecule then is cyclic adenosine monophosphate. Okay, cyclic AMP, or CAMP for short. Right, okay, so this is the reaction then that the adenylyl cyclase, once active, will catalyse. It will convert ATP to cyclic AMP and pyrophosphate. Okay, right, so it is these enzymes which the alpha I GTP subunit is famous for interacting with. Now, basically, there is not just one adenylyl cyclase enzyme, there are actually 9 different adenylyl cyclase enzymes, and these are called very sensible names. So they are called adenylyl cyclase 1 all the way up to adenylyl cyclase 9. Okay, now the adenylyl cyclases are grouped into 4 different groups. Okay, group 1, group 2, group 3, and group 4. Okay, and basically the alpha I GTP subunit is not going to act on all of these 9 different forms of adenylyl cyclase. Instead, it's going to act on adenylyl cyclases that are in certain groups, and we'll see the grouping of the adenylyl cyclase enzymes in the next video.